There we go. Live. And live. Excellent. We've both got them both going live. I think we're ready now. Hello. Welcome to Drunken Sexy Science. I'm Dr. Jessica Hebert, and I'm here tonight because I made a terrible deal. I made the deal that if we reached 75% on our Kickstarter backing last night, that I would do this. I would put up a poll today, and we would talk about what it is that you wanted to see me do on the internet that was a little bit ridiculous, a little bit silly. And I should not be surprised at all that of the three options, one was spin and swear. I have a Peloton bike that I love to pieces, and it kicks my butt on the regular. And I have a couple of favorite Peloton instructors. I love Matt Wilpers. He does a lot of the Power Zone training. And Dennis Morton, also he's really pretty. And Emma Lovewell is fantastic, and sometimes they make me swear a lot on the bike. So I was going to do a spin and swear, which still might happen. Option number 2 was to meet my dog Lucy, who is a big fluffy muppet, and she would get treats as I read her a story while you on the internet provided Kickstarter backing. And option number 3 is what we're doing right now. Welcome to Drunken Sexy Science with Dr. Jessica Hebert. So what that means is I'm going to tell you a little bit about how to read a scientific paper tonight, at least the way that I do it. And then I'm going to read you some excerpts from a really cool scientific paper and sexy as two meanings. It's not just the way that I'm reading it, but it's also going to be reproductive science. We're going to feed you some vegetables along with your candy. I hope that's okay. Eventually, if this goes really well, it's a thing that I've kind of wanted to do for a while. We might talk about doing some ASMR type recordings, like the sound of the warm towels and the clicking of different things, the sound of rain, but cupped around. Binaural recording is what it's called. We can talk about that maybe. If you find this soothing, we can talk about how to make it even more soothing so you get that stereo sound of science. So the chat boxes are open. Go ahead and say hello. Tell me what you're excited about in science. If you have questions about reproductive science, as long as they aren't going to violate HIPAA, I don't want to hear about your sores or anything in the box. I'm sorry, go see a doctor. But I'm happy to answer other questions about reproductive science. In addition, I have wine. I have an entire glass to start with because not only did you get us over 75% last night, we are currently sitting at 81%. So I thought it was more than fair for me to start with a glass. I also brought in the box. So for every $10 that our Kickstarter goes up this evening, I'll take a good sip of wine. Not like a little sip, like a good sip. And if you donate if it jumps more than $100 at once, I'll just finish the glass and pour myself another one. I plan to be doing this for about 45 minutes, but if you all make that Kickstarter number jump, it might be less time that I'll be able to sit upright. So, hi Tracy. Hello everybody in chat box land. Hello YouTube. The chat box is open. Feel free to say hello and ask your questions about sexy science. Box wine is good wine. It's Boda Box's Sauvignon Blanc. It's perfectly fine. I belong to a wine club, but I'm drinking box wine tonight because I have a feeling y'all going to get me a little bit schlizzard. Let's see where we are right now. The Kickstarter is at 10, 1, 95, 85, or 81%. If it was 85% already, I'd be in trouble. Oh, crap. We've already jumped. I haven't even done anything yet. I just turned on the video. Oh, Patty. Hello, Patty. And Patty's already making me do math. It's a $30 jump. So for Patty, 3 good sips. There's one. Deux. Trois. All right. We've already whined and we're like 30 seconds into this. You know that this is going to be a good party. All right. Pow, pow. So the way that I usually read a scientific paper, in case you were curious, if you are scared of reading scientific papers because you're like, that seems really hard. They seem really dense. I mean, some of them are. This is the one that I'll be reading a bit to you tonight. They can get pretty dense. So when I read hi Kathy, when I read a scientific paper for the first time, because I've read enough of them, if they're in my field, I will read the bit at the top first. It's called the abstract. If it's not in my field, or for you, if you don't read a lot of scientific papers, pick something that you're kind of excited about, you're interested in, whether that's archaeology or dinosaurs or microbiota or whatever that is. Find a paper, skip the first part. It's usually labeled abstract, and what that is a summary of everything that you're about to read. And sometimes it can get really dense and scary right away, because what they're telling you in the abstract is in 250 words generally. What are you reading about in terms of the big background field? Why is this paper really important? Some of their basic methods that they used to write this paper, their basic findings, and what they think that means. Hi, Kathy. So what all of that happens in 250 words, and that's overwhelming. So if it's your first time reading a scientific paper, skip the abstract. I would read the introduction. When you get to the end of the introduction, if there was something that you didn't quite understand, usually what happens is it starts slow and it starts to build near the end. And so if you missed something, take a look at the little numbers at the end and flip to the back, and those are references. And you can go, I didn't understand what this meant. And sometimes just looking up that reference and reading what that paper was about, you can go, okay, I kind of get it. Or you can actually go and look up that paper. But then after that, usually there's a methods section, depending on what paper you're reading. Skip the methods. Skip the methods. Go to the results and skim through the results and see, what is it that they found? What's the exciting part? Because if it's not exciting, if there isn't a reason to talk about it, they're not going to talk about it. The results is where you get that meat. The results is where you get the interesting stuff. So read through the results, look at the graphs, and see if you agree with what they said. Once you've kind of determined that, then read the discussion, and that's where they tell you what they think it means. So if you're reading a paper for the first time and it's not in your field, or you don't have a lot of experience reading scientific papers, skip the abstract, read the introduction, read the results, read the discussion. After that, if you want to go back, you can read the methods. How did they do that? And you can read the abstract, and by then it should make a little bit more sense. We tell our cell bio students to read each paper a minimum of 3 times. One, the way I just described, intro, results, discussion. The second time, you can read all the way through. You can read with the methodology sort of to get your brain going into what was it that they were thinking. And then the third time, read the abstract and see if you think that was an accurate summary of what you just read. Sexy science, it's facts. So if you're watching at home, feel free to say hi in the chat box. For every $10 that our Kickstarter goes up this evening, I'm going to take a drink of wine. I have my wine glass here, and I have this entire Boda box behind me, so I'm ready to go. Behind me, you see the wall of SciComm. I've got Carl Sagan, Bill Nye, and Neil deGrasse Tyson. Right behind my head is Rosalind Franklin. Rosalind Franklin is my favorite. She's my superhero. And if I read a book to Lucy, perhaps tomorrow or sometime this week, since that was the second ranking thing, it will probably be Eureka by Danault and Ashley Hammond, because Lucy likes Eureka and it's all about scientists. And I can read what I want on the internet. Right, I'm supposed to be drinking when you pledge. This is the problem. The wine is really good. Hello, Dr. Epstein. It's good to see you, Dr. Epstein. So I'm checking our Kickstarter levels again real quick before I jump back in. So we're at 10, 2, 2, 6. So I'm not going to drink any more right now because you haven't made me. Except I clicked some buttons and now I'm in trouble. Hold on, hold that thought. I don't know what I just did, but I'm sure it was terrible and ill-advised. That's kind of the story of my life. Did you know terrible and ill-advised is the Jessica Hebert story? Pew pew. That's not what I want to do at all. I want that. I want that. Okay, great. Go ahead and use the chat. But for right now, I'm going to begin reading a paper by Kirsty Agard, June Ma, Kathleen M. Antony, Radhika Ganu, Joseph Petrosino, and James Versalovic. They're all from Baylor University in Houston, Texas. And I really like this paper, which is called The Placenta Harbors a Unique Microbiome. I like this paper because it was one of the first big research articles to talk about the microbiome, which is all of the bacteria and microorganisms that live in the placenta. It's not a sterile environment. And a lot of folks think that in the womb things are sterile, and they're really not. There's all sorts of things growing in there. And while the baby is considered in a sterile environment as long as the amniotic sac is intact, once the baby passes through the vaginal canal, baby is definitely contaminated, and baby can be exposed to microorganisms living in the placenta. It's not as sterile as we thought. What this paper says is you've got a lot of different colonies of microorganisms living in your body, in your mouth, in your intestines, on your skin. And what they're saying is the microbiome that inhabits the placenta is different than the microorganisms that you might find anywhere else in the body. Though, surprisingly, they are most related to the microorganisms you find in mom's mouth, not in her intestines, not in her skin, not anywhere else. And that was very surprising to find, especially since we know that the microbiome of mom's mouth, whether she's obese or lean or she has a disease like diabetes, that microbiome changes, and so does the one of the placenta to sort of match. So I'm going to read you, I'm going to skip the abstract for right now, but I'm going to read you the introduction. And then I'm going to go back and read you the abstract because we're a little bit limited on time and also vocal ability this evening, so that you understand what is this paper about and then a brief summary of their findings. Let's begin. The placenta harbors a unique microbiome. Senior investigator is James Versalovic, again from Baylor University in Houston, Texas. Introduction. Over the past 4 million or so years, hominids have co-evolved with their microbiomes as physiologic communities composed of distinct body site niches. Recently published works by the Human Microbiome Project Consortium have provided the first reliable estimates of the breadth of structure, function, and diversity of the healthy reference human microbiome across multiple body sites. Despite recent advances in cataloging the complexity of the adult human microbiome, there are limited population-based data as to how and when the human microbiome was established. Characterizing the processes that govern the establishment of human microbial communities is essential for understanding human development and physiology. Studies have shown that neonates demonstrate complex microbial communities in the gut within the first week of life, with dynamic fluctuations in bacterial composition until a relatively mature equilibrium is reached around 1 to 3 years of age. What shapes these earliest microbial communities, and what point does the infant first exposed to and colonized by its microbiome remains unclear. In the first week of life, the full-term neonatal gut microbiome is largely colonized by the phyla actinobacteria, including bifidobacterium, proteobacteria, bacteroides, and much less firmicutes, including the lactobacillus subspecies, which dominate the vaginal flora. In contrast, neonates who weigh less than 1,200 grams, 12,000 grams would be a very big baby, 1,200 grams, 1.2 kilograms, is about 3 pounds. Neonates who weigh less than 1,200 grams are dominated by both firmicutes and tenericutes phyla, with much less dominance of actinobacteria. These collective observations raise the possibility that the infant may be first seeded in utero by a common shared low abundance source, such as the placenta, and that this seeding may vary by length of gestation. Establishment and maintenance of placental integrity and function are critical to fetal growth, development, and survival. Previous studies have largely used morphologic approaches and grossly documented the presence of bacteria in different regions of the placenta. A recent cross-sectional study of 195 patients demonstrated gram-positive and gram-negative intracellular bacteria harbored in the basal plate, which compromises the tissue layer directly at and below the maternal-fetal interface. These were observed in nearly one-third of placental specimens, with a high prevalence among preterm deliveries less than 28 weeks of gestation, but regardless of clinical or pathological evidence of chorioamnitis. Chorioamnionitis. We say it a lot, and I totally tripped. Chorioamnionitis means that there isn't enough fluid around the baby. Although current paradigms suggest that most intrauterine interactions, which are associated with preterm birth, originate in the lower genital tract and ascend into an otherwise sterile intrauterine environment, most taxa detected in the placenta with DNA-based technology are not found in the urogenital tract, but rather represent commensal species common to the oral cavity. Some of these oral microbiomes, such as Fusobacterium nucleatum, a gram-negative oral anaerobe, that means it grows without oxygen, may facilitate hematogenous transmission during placentation, blood transmission, as a result of their ability to bind vascular endothelium and alter permeability, thereby functioning as an enabler for other common commensals, such as E. coli. These latter findings are of particular interest given the long-standing strong association between periodontal disease, as well as upper and lower urinary tract infections, and increased risk of preterm birth. Here we have leveraged recent approaches and tools for metagenomic analysis to characterize a rigorously collected set of placental specimens from 320 subjects in a case cohort design. We present here an initial snapshot of the human placental microbiome and reveal which organisms are present, what they are capable of doing, and how the placental community is likely structured. So that was the introduction to The Placenta Harbors a Unique Microbiome by Versalovic et al. From Baylor University in Houston, Texas. So let's talk about what that means. There were a lot of words in there. And if you have never done any sort of placental research, you go, that was a lot. I do a lot of placental research, and it was still a lot. What they were saying was they discovered that if baby is born normally, there is one set of bacteria that dominate the placental environment, whereas if baby is born very small, 1.2 kilograms or less, that it's actually flipped. The microbiota, the little bacteria that you would expect to dominate, are actually the smallest population, and the previous smallest population are now the largest population. Moreover, they found that there's a connection between oral disease in the mother and issues with baby's size and the microbiota that you see in the placenta, which again lends credence to the idea that it's possible that the microbiome of the placenta matches what you would see in the mouth and not anywhere else. So that's pretty exciting. They go on in the rest of this paper they do metagenomic analysis, which means they take the entire sequence of DNA and they slice it up, and they, I believe they did, I need to look at the methods on this one. I believe they did a chip on chip. I will find out and let you know. Essentially, they chopped up the DNA and ran it through a sequencer and said, what's here? What's here? Let's line it all up. You've got these little tags, and then you can make these big sequences. And they looked very specifically at this ribosomal subunit called 16S that we all have. And they said, or that all of these different microorganisms have. And they compared them to all the different ribosomal segments of known bacteria and said, okay, so here's what's here. Does it match what we already know? We have a database for this through the National Institutes of Health, which you can go and you can look up the sequence of anything you've ever wanted to know about. So they compared it against this big database of sequences, and they said, all right, what we actually found was true. The microbiome of the placenta matches what you see in the mouth. It is unique of itself, but it is most closely representative of oral microbiome and not anywhere else. So that's pretty exciting. I'm going to read the abstract to you in a minute just to reemphasize that, but I'm going to check our Kickstarter levels right now to make sure if I need to be drinking. I'm going to be drinking. If you're just tuning in, for every $10 donation to our Kickstarter this evening, which you can find at bit.ly/relatable broads, capital R, capital B, I will take a drink of wine. And if you donate, if the number jumps $100 or more at once, I will drain the glass and start anew. So it's a good deal for you. It's a good deal for me. I have an entire box of wine and about another 30 minutes or so to go. And I've got a couple more papers that I can read to you. I'm also happy to answer any of your science questions, particularly sexy science, as long as they don't violate HIPAA. Yes, that's a rash. It will be $400. I'm not that kind of doctor. It's true. I have a PhD, not an MD. Though my PhD is in STEM, it's in biology, and my focus, for those of you who don't know, is the placenta, which is why I'm telling you all about placental microbiome. In particular, I look at obese versus lean pregnancies and the effects of gestational diabetes. Previously, I looked at preeclampsia. I've also done work in HPV and vestibulitis and a number of other things. I don't know what the little camera up in the corner means. Am I supposed to do anything with it? I hope not. No. Good. Somebody else is apparently live streaming at the same time. They said, would you like to bring them on with you? I don't think he wants me to bring him on. Hi, Shane. Unless you want to talk about placentas with me, in which case I'd be delighted to have you. So we are currently at 10:26, and I'm ready for more wine. So please feel free to donate to our Kickstarter, which again, you can find by searching on Kickstarter for the PDX Broadsides, or also bit.ly/relatable broads with capital R and capital B. One more refresh real quick, because the last thing I want is for it to jump $100 all at once when I wasn't paying attention to it, because then I have to drink. All right, so this is fun, isn't it? Reading papers and drinking wine. That's how we do. So that was the introduction to the placenta harbors a unique microbiome. Now I'm going to read you the abstract to that paper, same voice, so that you can hear the way that the introduction now wraps into what it is that they found and their overall conclusions from that paper. Humans and their microbiomes have co-evolved as a physiologic community composed of distinct body site niches with metabolic and antigenic diversity. The placental microbiome has not been robustly interrogated, despite recent demonstrations of intracellular bacteria with diverse metabolic and immune regulatory functions. A population-based cohort of placental specimens collected under sterile conditions from 320 subjects with extensive clinical data was established for comparative 16S ribosomal DNA-based and whole genome shotgun (WGS) metagenomic studies. Identified taxa and their gene carriage patterns were compared to other body site niches, including the oral, skin, airway, nasal, vaginal, and gut microbiomes from non-pregnant controls. We characterized a unique placental microbiome niche composed of non-pathogenic commensal microbiota from the firmicutes, tenericutes, proteobacteria, Bacteroides, and Fusobacteria phyla. In aggregate, the placental microbiome profiles were most akin Bray-Curtis dissimilarity less than 0.3 to the human oral microbiome. 16S-based operational taxonomic unit analyses revealed associations of the placental microbiome with a remote history of antenatal infection, permutational multivariate analysis of variance p equals 0.006, such as urinary tract infection in the first trimester, as well as with preterm birth less than 37 weeks, p equals 0.001. What does that mean? That means, again, as they addressed in their introduction, they took these placentas and they said, what kind of microbiota are in here? We're going to look at their 16S sequences, comparing the different kinds of microorganisms from different areas of the body, from the vagina, from nasal passageways, from the skin, from the gut, and say, what is it most similar to? And they said that it is unique, but it is most similar to the human oral microbiome when they lined everything up. And that there were certain things that they could tell from this microbiome that shifted, including having a urinary tract infection in your first trimester or being born too early. So all of those things they could tell from just looking at the microbiota of the placenta. That's pretty neat. So it looks like we have a bunch of questions via Twitter. They're not coming through the chat. So I'm going to try to pull up Twitter separately. So many windows to make things work appropriately. If you're online and you're watching and you have sexy questions, sexy science questions, especially reproductive science questions, I'm here for you. As Cody says on Peloton, I got you, boo. Get your life together. Lots of people apparently live streaming it on Twitter. That's great. I can't see if you're making comments for some reason, they're not popping up on the chat dashboard through Restream. Restream is the way that I'm doing this tonight for YouTube, Periscope, and Twitch. Hi, Twitch. This is our very first Twitch stream. We've never tried this before. I'm also doing a live cast on Facebook, but I'm doing that separately because the only way to get Restream to do Facebook is to stream it to Christian's page right now, and I don't think he would like that if I was talking about the placenta on his page for an hour. He gets to deal with that with me in the car on tour all the time, and hopefully he'll have to deal with me in the car on tour on the East Coast very, very soon, because we are so close. We are within 20% of our goal to make relatable content, the album and tour, not just a really fantastic album of 13 songs about feelings and love and heartbreak and science and hashtag me too, but also how much we really love brunch and why riding buffalo is bad for your health, bad choices. So we have a lot of great music on this album that we're excited to share with you. And our East Coast tour, we've already booked 4 stops, and we would like to tell them that we are still coming and not bail. and we've got several others in the works, and we would like to come. We've got friends that we would like to sing with, including the Misbehaving Maidens, who closed out their massively successful Kickstarter last night. They funded at just over $20,000. They get to make their album and make a video with our label parents, Laser Milena, our label parent, Laser Milena Weber of the Double Clicks. And yeah, they get to do all sorts of neat stuff because Kickstarter and this platform where you all believe in us, and we really appreciate that. So again, if you haven't been to our Kickstarter yet, why? It's really great. Don't watch the video right now, you're watching me, but the video is super duper cute. It's bit.ly/relatablebroads with an R and a B capitalized. You can also just search on Kickstarter for relatable content, PDX Broadsides, or find us on Facebook or Twitter, where we've been talking endlessly about this Kickstarter now for several weeks because we are stoked about it. We are still at 10:26. I'm still going to finish this glass of wine anyway because I like wine and I like you. So this is for everybody who got us over that 75% hump, and you've believed in us every step of the way, and we are very, very grateful. So we've talked about the placental microbiome. Let's talk about a different aspect of the placenta. I'm just trying to decide which aspect we should talk about. Now, these papers, I'm telling you, I've never read any of these papers out loud before, so some of the words are exciting, and sometimes I'm discovering new things about, like, even in that paper, as I was reading, I went, oh, right, they did WGA, whole genome analysis, and I didn't think about it until I reread that bit in the abstract. Right. Let's talk about humanin. Humanin? Insulin. Insulin and metformin. That's what we're going to talk about. Welcome to Drunken Sexy Science with the PDX Broadsides. I'm Dr. Jessica Hebert. Please don't ask me about your sores. That'll be $400. It is an infection, and I'm not that kind of doctor. Not legal medical advice. Ooh, don't take legal medical advice from ponies who live in sheds. That's just good advice. I need more wine. Here's where we are. So I've just read you a paper, an introduction abstract about how the placenta has this unique microbiome. If you are curious about that paper, again, the senior author on that was James Versalovic from Baylor in Texas. What I'm going to read for you now, starting again with the introduction, and then we'll discuss it a little bit, and then I'll move back through the abstract so that you don't get overwhelmed if you're not super familiar with placenta science. We're going to talk about the placenta and insulin, which is an important thing to understand when you are looking at gestational diabetic pregnancies. Gestational diabetes is a diabetes that only happens when you're pregnant. There are two types, just like there are two types of regular diabetes, type 1 and type 2. Type 1 is when you can control it with diet and exercise. Type 2 is when you have to treat it with insulin. It's actually kind of a little bit backwards from what you would expect. So type 2 gestational diabetes is of particular interest to me, and we treat these women with insulin. But it has been recently found that insulin might be toxic to the placenta. So they've started treating them with a second drug called metformin. If you are an older white male with high blood pressure and diabetes, you might be familiar with metformin. Metformin is used to control diabetes in a number of patients, but particularly older white men for some reason. So I'm going to talk about this paper, which is called Direct Toxicity of Insulin on the Human Placenta and Protection by Metformin, where they treated the placenta with both insulin and metformin together to see if metformin had a protective effect on the placenta against insulin. This paper is by Mario Vega, Maurizio Mauro, and Zev Williams. Doctors Vega and Mauro are from a university. I'm sure they are. There that is. The Albert Einstein College of Medicine, Yeshiva University in the Bronx. And all three, Doctors Vega, Mauro, and Williams, are from Columbia University in New York, New York. Introduction. Insulin resistance, defined as impaired metabolic response to insulin, affects 33.9% of the U.S. population, and that's 84.1 million people, and is markedly increased by a risk of poor pregnancy outcomes, including fetal anomalies, delayed development, perinatal morbidity and mortality, and recurrent miscarriage. Although the pathogenic mechanism is not currently known, hyperglycemia has been the presumed etiologic agent, and screening and treatment efforts have been focused on detecting and preventing hyperglycemia. Insulin resistance differs from type 2 diabetes mellitus in that although insulin levels are elevated, glucose levels remain normal. Data from animal and clinical studies suggest that insulin itself may play a direct pathogenic role in pregnancy. For example, murine models of hyperinsulinemia have increased rates of embryopathy and apoptosis in blastocysts, even in the setting of euglycemia. Clinically, women with type 2 diabetes mellitus, characterized primarily by hyperinsulinemia, have worse perinatal outcomes than women with type 1 diabetes mellitus, characterized primarily by hyperglycemia. Although insulin has traditionally been categorized as an anti-apoptotic and pro-proliferative growth factor, the direct effect of insulin on primary human trophoblast cells has not been previously studied, particularly at concentrations found in women with insulin resistance. To determine the direct effect of insulin on the early placenta, we collected first trimester human trophoblasts after elective terminations of healthy pregnancies at 6 to 8 weeks of gestation and cultured them for 48 hours in media supplemented with either vehicle control or insulin concentrations, simulating an in vivo concentration found in patients with insulin resistance. These were assayed for the effect of DNA damage, cell survival, and apoptosis. So that's the introduction to this paper. Hi, Patty. The current drink count is, it was 3 good sips, but I finished this glass of wine, and I'm about to pour myself another. Hopefully the count goes up. Let me recap that introduction for you real quick, even though the wine is starting to swim my head. What has been thought is that there's insulin resistance in these pregnancies. It's been assumed that having too much sugar in your blood is what causes it. But it turns out that type 2 gestational diabetes, there's an insulin resistance. What they did was they took placental cells from first trimester, human placentas that were obtained from elective terminations, that means elective abortion, and then they cultured those cells for 48 hours and gave them either insulin or not insulin to determine the way that these placental cells grew very early in pregnancy, and to see if by treating them with insulin and metformin or just insulin, if that had a effect on the way that the placental cells thrived or died, and if they could have a protective effect using metformin. And I'm guessing from the title of the paper that they did. They did. Spoilers. I know. If you're planning on reading this, early spoiler, but the graphs are always really good to look at. In fact, they have this beautiful figure right away, figure 1, that tells you exactly how they plated all of their cell cultures. So that's pretty great. To get to see methods in pictures, I do like that because it does help you put into context exactly what it was that you did, rather than just a graph and then some words that explain that. That's pretty helpful. Patty says, this is a better paper than the public health ones I'm currently reading. It's a pretty solid paper, I won't lie. And it helped encourage me to look into a couple of additional drugs to treat my placentas, and currently that's going very, very well. So that's why you read literature, because it helps stimulate ideas, and you kind of form up your own ideas about the direction that you would like your research to go. So the drink count right now is 3 good sips, although I did finish my glass of wine because I love you and you have done so well. And I'm just checking that number right now, and it hasn't moved. We did top 10,000 today, which is remarkable. So we are currently at 10,226. And for every $10 that number goes up, I'll have a good sip of wine. And if that number jumps more than $100, I will finish the glass and pour another one because, friends, you are worth it. So I've got probably about another 15 minutes or so in me of paper reading and explaining, and I'm happy to take your questions while we are chatting here together before I'm going to probably not polish off this entire box of wine. But I will do this again probably tomorrow night, either this or the second most popular option on the poll today, which was to read a bedtime story to Lucy. For every $10 you donate, Lucy will get a little kibble covered in peanut butter. And if you haven't met my dog before, she is the best. She is a 70-pound Muppet and the best dog in the history of ever. So is your dog. Your dogs are also the best dog in the history of ever. So all of our dogs can be the best dog in the history of ever. Dogs are so good. They're so good. Bront. All right. Again, if you're just tuning in, the Kickstarter is bit.ly/relatablebroads. And that R and that B are capitalized. If you can't see it right now, I am wearing a shirt that says Schrodinger's cat is dead. My father got very angry about it earlier when I went to go see my mom for Mother's Day, which is why I'm a little bit late on. He says, "The cat's not dead." On the back of the shirt it says, Schrodinger's cat is not dead. It's that kind of shirt. That's how we go. More pics, less words. Yeah, that'd be gross, except it's my cousin, which just makes it double gross, doesn't it? Hi, Andy. I'm going to refill this wine, and then we're going to recap this paper. Go ahead and ask me any questions about reproductive or sexy science. Yes, it's infected. No, you shouldn't have slept with her, and that will be $400. All right, I have a new glass of wine. I took a sip because that one was for free and for me. All right, again, the introduction you just heard a few minutes ago was from Direct Toxicity of Insulin on the Human Placenta and Protection by Metformin from Drs. Vega, Morrow, and Zev Williams. The senior author is Dr. Zev Williams from Columbia University. Drs. Vega and Dr. Morrow are from the Albert Einstein College of Medicine, Yeshiva University in the Bronx. The abstract, as we just talked about just a little bit ago, if you're just tuning in, usually when you read a scientific paper, I advise that you first read the introduction and then the results so that you sort of get into your own head what you think this paper is about, and then read the discussion. If you aren't familiar with the subject area, skip the abstract on top at first, go back to it later because it will often just overwhelm you or make you start making conclusions before you actually have read the rest of the paper. So make sure that you have a good idea of what's going on before you let yourself be swayed. So again, this is the abstract to Direct Toxicity of Insulin on the Human Placenta and Protection by Metformin. Objective: to study the effects of insulin metformin on primary trophoblasts from early pregnancies. This is an experimental in vitro study at an academic research institute. The patients are trophoblasts from healthy patients undergoing first trimester elective termination of pregnancy and primary lung fibroblasts. The intervention proposed is culture and treatment with insulin and metformin of primary trophoblasts and primary lung trophoblasts. Main outcome measure: DNA damage measured by expression of gamma H2AX with immunofluorescence and Western blot. Apoptosis measured by expression of cleaved caspase-3 by Western blot. Cell survival measured by cell proliferation assay. Result: culture of purified primary trophoblast cells in the presence of insulin at levels as low as 1 nanomolar resulted in a 386% increase in the number of cells with elevated gamma H2AX expression, a 66% reduction in cell survival, and a marked increase of cleaved caspase-3 expression. Pre-treatment of trophoblasts with therapeutic doses of metformin prevented the detrimental effects of insulin. Treatment with insulin and/or metformin had no effects on primary fibroblasts. Conclusion: elevated insulin levels are directly toxic to first trimester trophoblasts and result in increased DNA damage, apoptosis, and decreased cell survival. These effects are prevented by metformin. Trophoblast cells from early pregnancy are uniquely vulnerable to elevated levels of insulin. These findings, if confirmed in vivo, suggest that there may be a role for insulin resistance screening before attempting pregnancy and for focusing on prevention of hyperinsulinemia during early pregnancy. A lot of words in the abstract. Cool finding. The cool finding is that when they treated the placenta with insulin alone without metformin, they found a huge increase in the number of cells with gamma H2AX expression, so increased inflammation, a 66% reduction in cell survival, so the cells were dying, and a marked increase in cleaved caspase 3. But when they treated them with metformin before they treated with insulin, they saw a decrease in all these detrimental effects. When they treated with insulin and/or metformin on primary fibroblasts from the lung, just to show as a control, they found no difference. So it's the placenta specifically that's responding to insulin and metformin. Hi, Shelby. Shelby says, one perk I'm seeing with genetics as a field of research, beautiful methodological, I've been drinking, charts. The rhetoric texts that I'm reading, sadly few illustrations. Unrelated, I'm loving the wall art. Oh, thank you. This is my wall of SciComm. Sagan, Nye, Neil deGrasse Tyson, Rosalind Franklin. I've got a lot more Rosalind Franklin art, including across my office, there is a Rosalind Franklin bobblehead. If our donations go up $20, I'll get up and show you the Rosalind Franklin bobblehead. It'll be funny to watch me try to get up because I've had now just over a glass of wine in 40 minutes. So that's been exciting. So, insulin and metformin. Ooh, this is fun, right? The upshot of that paper is if you're pregnant and you're taking insulin, you should take metformin too, or else you're going to damage the placenta. And a damaged placenta leads to a smaller baby. And those babies end up growing up to have health problems at a higher rate, like gestation, like diabetes, kidney failure, heart failure, cardiovascular issues, high blood pressure. And we want those babies to have the very best chance at growing up to be healthy adults as possible. So that's the work I do. Healthy mamas, healthy babies. And I have a very, very powerful belief that if we can start treating gestational diseases now, like gestational diabetes, preeclampsia, maternal hypertension, maternal drug use, we can make our world a lot healthier for those babies as they become adults. We can reduce the incidence of obesity. We can reduce the incidence of cardiovascular failure. And hopefully those babies will grow up to die of things that are much more exciting, like Ebola. So that was direct toxicity of insulin on the human placenta and protection by metformin by Williams et al. So 2 papers down. Again, if you're reading a paper for the first time, I highly suggest starting with the introduction and letting that go in your brain meats before you do anything else. Brain meats is a technical term. I learned it in a science. We are at 10:26. Great. So, and I don't have any questions on the chat or on Twitter. So I presume that people are just here to listen to me, which is fine. It's the exact opposite of what happens in the lab or in the band or in my real life. Ryan Taylor, yes, let people die by more exciting ways, like pirates. Well, maybe. Possibly, depending on where you are and how drunk they are. As you do. It's good wine, y'all. Box wine is good wine. Call me Boda. All right, I'm probably going to read you one more introduction this evening, and then I'm going to let you get back to your lives. But the PDX Broadsides relatable content, I can't even say it anymore, relatable content Kickstarter runs now through May 23rd. You can find us at bit.ly/relatablebroads, capitalize both words, also pdxbroadsides.com, and we're always on Twitter and Facebook and all the places, and we're not going away once this Kickstarter is done. In fact, I've had a lot of fun doing these live stream things, so I mean, there might be more. Who knows? Right now, it's a really good opportunity for you to get to know my science. I can talk to you a little bit and hopefully encourage you to go look at that Kickstarter. Patty asks, are you presenting anything public in Research Week this week? It is OHSU Research Week. That is where I'm a postdoctoral fellow. I am not presenting this year because I just completed my PhD in June, and I've been working in this new lab for less than a year. I don't have data that I feel really solid about presenting. I've got some good pilot results, but I am going to be judging the Tuesday morning surgery orals. So if you're interested in seeing me at Portland State, at OHSU's Research Week, come and join me Tuesday morning at 9 a.m. I think it's in the old library, which is their current auditorium, for the surgical discussions. There's also several poster sessions, and they'll have like liquid nitrogen ice cream and stuff at the poster sessions. Are always really good. Come and learn about the wonderful research happening at OHSU all week. OHSUResearchWeek.com. I don't know that that's true. I just made that up, but you can probably find it. One more paper with a good abstract introduction. Then I'm going to let you go for the evening. Well, this one's got a pretty picture on the front. Oh, good. Yeah, I'm actually getting, this is a bonus for me. I'm reading some papers that I was supposed to read that have been sitting on my to-read paper pile for forever, and now you're making me actually engage with them a little bit better, except for the placental microbiome one. That is one that I've loved for a very long time. But these are ones that I've been meaning to read, and now I am because of you. So thank you. Oh, Patty's got the link in the chat. For those of you who are watching via Restream, it's ohsu.edu/research-week. ohsu.edu/research-week. Come and see all the wonderful research happening there. Patty, are you presenting? Are you? While we wait for Patty's response, because there is a delay in these things, I'm going to read you the introduction, and then we'll do a brief chat back, and then the abstract. 2. Placental polyamine metabolism differs by fetal sex, fetal growth restriction, and preeclampsia. This paper has a ton of authors on them. Gong, Sovio, I, Gacchioli, Doppler-Alla, Johnson, Wood, Cook, Jenkins, Coleman, Casero, Constancia, Charnock-Jones, and Smith. And I know the senior investigators on this, Stephen Charnock-Jones and Gordon Smith. Most of them come from the Department of OBGYN at NIHR Cambridge, or the Center of Trophoblast Research, again at Cambridge. And it looks like most of them come from Cambridge, with the exception of, where is the 5? Dr. Casero, who is at Johns Hopkins. Patty is not going to be presenting at OHSU Research Week, so you're just going to have to deal with me judging and my judge face. So, placental polyamine metabolism differs by fetal sex, fetal growth restriction, and preeclampsia. This is the introduction by Gong et al. Preeclampsia and FGR are two of the great obstetrical syndromes. These disorders cause short-term complications for the infant and have been associated with a range of diseases in adult life, such as ischemic heart disease, stroke, type 2 diabetes, and long-term neurodevelopmental disorders. Moreover, these complications have profound effects on maternal morbidity and mortality. Preeclampsia is one of the leading causes of maternal death globally, and both preeclampsia and FGR are markers for the mother's later risk of cardiovascular disease. Both preeclampsia and FGR are associated with abnormal placental function and metabolism. Currently, the placental mechanisms leading to preeclampsia and FGR are only partially understood. It is also unknown why placental dysfunction can lead to preeclampsia without fetal growth restriction in one woman and fetal growth restriction without preeclampsia in another. Epidemiological studies have demonstrated that fetal sex is associated with different patterns of ultrasonic markers of placentation, placental pathology, the risk of perinatal death, and the risk of preeclampsia, but the mechanisms underlying these differences are unknown. Given these observations, we hypothesize that the placenta would exhibit sex-related differences across multiomic analyses. Moreover, we hypothesize that sex-related differences in placental function would be associated with the risk of placentally related complications of human pregnancy. We tested these hypotheses using 3 discovery-based methods, namely analysis of the placental transcriptome, the placental methylome, and the mother's serum metabolome. We then used targeted in vitro experiments in primary cultures of human trophoblast cells to confirm sex-related differences in function. So we'll talk about that in half a second. Patty says 2 members of her cohort, she's a student at OHSU, are presenting and they are fantastic. So if you're coming to OHSU Research Week, look for Nate Evans and Gareth Harmon. So the introduction, what does this mean? Preeclampsia and FGR is what they're talking about primarily. So FGR stands for fetal growth restriction, means a small baby compared to placental size. It's all about placental efficiency. Fetal growth restriction is anytime you've got a small baby. Preeclampsia is the failure of the placenta to pass the appropriate amount of nutrients and blood from mama to baby. It's an inefficient placenta. In preeclampsia, you also see mom's blood pressure stays high throughout pregnancy. Her blood vessels near the placenta don't twist right to slow the blood flow down between mama and baby. You get really bad swelling. Preeclampsia is no good. But they are finding that there are pregnancies. They say women a lot. I'm trying to say folks more because you can be pregnant and not female. That you can have preeclampsia and not have a small baby, or you can have a small baby and not have preeclampsia, and you're not sure why these things happen when they happen, because they are often comorbid. They don't necessarily have to be. So they wanted to understand what the markers of that were. So they took the placenta and they looked at the transcriptome first. The transcriptome is all of the gene expression that comes from the placenta. What's being expressed? That's a transcriptome. 2, the methylome. This is a thing called epigenetics. So not just what's on your DNA, but what's on the outside of your DNA is really important. And the things that are on the outside of your DNA that affect the way things are expressed. This is part of the methylome, epigenetics. What's on the outside of your DNA causing things to be expressed. So the more methylation you have, in general, the less expression you have. Methylation usually turns off a gene. Sometimes it doesn't. So they looked at methylation patterns to see if there was something in the environment that caused genes to turn off by affecting the outside of the DNA. So gene expression, methylome, and the third thing they looked at was the mother's serum metabolome. So they took blood from mom, they spun off the serum, so red blood cells go down, the serum goes up. They took that serum and they said, okay, what's in this, metabolically speaking? So they compared all of those things, and they don't say in the introduction what they found, but I bet they do in the abstract. So I'll read you the abstract in just a moment, as soon as I check our Kickstarter to see if we've jumped at all and if I need to drink more wine, which I'm getting out some words, but some words are coming out a little bit chunky, friends. Nope, I don't. We are at 10:2:26. I'm going to have a little bit of soda water instead. I came prepared. I'm just saying, you want me to drink more wine? It's going to cost you. Or it's going to cost me one way or the other. Woo! So, here's the abstract. Again on that paper, this is placental polyamine metabolism differs by fetal sex, fetal growth restriction, preeclampsia by Gong et al. Oh, fetal sex, that's the other thing. So remember how I said when you have a problem in pregnancy, it can affect the way that the baby has adult diseases long term? Yeah, I left something out on purpose, and now I'm bringing it back in. It's worse when baby is a boy. That's right. And we don't fully understand why. We have some ideas, but I'm not going to get into them tonight. But boys do worse than girls. There is a great paper by Kent Thornburg and a bunch of other folks. It is one of my favorite named papers in the entire field. It's called Boys Live Dangerously in the Womb. It is worth you checking out. So here is the abstract on placental polyamine metabolism differs by fetal sex, fetal growth restriction, and preeclampsia. Preeclampsia and fetal growth restriction are major causes of more than 5 million perinatal and infant deaths occurring globally each year, and both are associated with placental dysfunction. The risk of perinatal and infant death is greater in males, but the mechanisms are unclear. We studied data and biological samples from the Pregnancy Outcome Prediction Study, a prospective cohort study that followed 4,212 women having first pregnancies from their dating ultrasound scan through delivery. We tested the hypothesis that fetal sex would be associated with altered placental function using multiomic and targeted analyses. We found that spermine synthase escapes X chromosome inactivation in the placenta and is expressed at lower levels in male primary trophoblast cells, and male cells were more sensitive to polyamine depletion. The spermine metabolite N1-N12 diacetylspermine was higher in the female placenta and in the serum of women pregnant with a female fetus. Higher maternal serum levels of diacetylspermine increase the risk of preeclampsia but decrease the risk of fetal growth restriction. To our knowledge, diacetylspermine is the first maternal biomarker to demonstrate opposite associations with preeclampsia and FGR, and this is the first evidence to implicate polyamine metabolism in sex-related differences in placentally related complications of human pregnancy. What does that mean? What that means is when baby is a boy or when baby is a girl, there is more of this particular metabolite in mom's blood versus when the baby is a boy. These higher maternal serum levels increase the risk of preeclampsia, but decrease the risk of fetal growth restriction. So it's the first time that they've been able to say that there might be a difference in something released from the placenta in mom's blood that would be able to tell about risk factors for preeclampsia and FGR and that difference between the sexes. So that's kind of exciting. Hi, Steve. Sex-based differences in the placenta, being able to measure biomarkers in maternal serum and placental cells by looking at the methylome and the transcriptome. And this paper will go into more detail, showing the exact percentages and layout of what you see in male versus female placentas and levels of expression. Oh, they've got a heat map. Heat maps are my former boss's most favorite thing in the entire world. So they found some very interesting things comparing boys and girls and looking at the placenta and mom's serum. Again, if you're interested in looking up any of the papers that I introduced to you tonight, we read the abstracts and the introductions too. The placenta harbors a unique microbiome, all about the weird and wild and wonderful zoological benefits that live in the placenta, and that's by Kirsty Agard et al., and it was published in Science Translational Medicine. Placental polyamine metabolism differs by fetal sex. Fetal growth restriction and preeclampsia by Gong et al. Was published in the JCI Insight in 2018. And direct toxicity of insulin on the human placenta and protection by metformin by Vega et al. Was published in Fertility and Sterility in 2019. It was one of the first articles published in 2019, and you can find that online. All these things are online. If you can't get them yourself at home, you can go to your local library. Your library will often have subscriptions to these journals. If you can't get them through your own library, you can ask for an interlibrary loan and contact OHSU or other local libraries. More journals are allowing certain papers to be open access. You can also go to ResearchGate and find those specific scientists and ask for a copy of their paper. We are allowed to distribute individual copies. We just don't own the rights to the things that are put out by journals. But if you contact us privately and ask us for a copy of a paper, we can do that. We're always happy to give you our own work. So that's a little bit of sexy science, a little less drunken than I hoped. I'm going to check Kickstarter one more time to see if we went up at all. For every $10 we went up this evening, I was taking a good old drink of wine. I did take 3 or 4 good drinks of wine tonight, so we're good to go. We are at 10,226. We broke the 10,000 barrier today. We're at 81%. We've got 19% to go. And if you're curious, yeah, we do have stretch goals. We'd love to tell you about them, but we have to fund the All-Women Tour first before we can move on. So please tell everybody that you love and some people that you don't love about the wonders of nerd music, about fandom and feelings, love and science, Buffalo, Steven Universe, and Arya Stark not having one more minute of your bullshit. All of those things and more coming up on Relatable Content. I'm Dr. Jessica Hebert. I am a postdoctoral fellow and a placentologist, as well as a singer-songwriter and pianist with the PDX Broadsides. It's been a joy to get to talk to you this evening. You can always find me on Twitter at PDX Broadsides or at Dame_DNA, or you can also look up hashtag Team Placenta. I'm always happy to chat science with you. Okay. Thank you for tuning in, and hopefully I'll have something for you again tomorrow night. Have a great night, and I wish you all a wonderful rest of your weekend. Bye.